In this video, I'm going to introduce you to Drosophila melanogaster and cover the basics of how to work with fruit flies in a laboratory setting. Fruit flies are an incredibly powerful model organism and can be used to answer a huge variety of questions. There's a large number of advantages to using Drosophila melanogaster, from their short generation time, going from egg to adult in as little as 10 days, to their easy maintenance, which requires a pretty minimal amount of work per week to take care of even a large population, to their clear sexual dimorphism, which makes it very easy to separate males from females, to their comparatively small and simple genome, and the vast toolbox of genetic resources that comes with it, which makes it incredibly easy to do complex or extensive genetic or genomic research with fruit flies. Fruit flies only have 4 chromosomes, which across them contain about 139 billion base pairs. Of these 4 chromosomes, only 3 of them really matter, being the X and Y chromosomes, the second chromosome, and the third chromosome. As you can see from the karyotype, the 4th chromosome is quite small and is usually ignored. Like most animals, fruit flies have XY sex determination, although their sex determination works a little bit different than other animals. Instead of determining whether or not an organism is male, the Y chromosome instead only contains genes that are used in spermatogenesis. Instead, for fruit flies, sex is determined by the ratio of X chromosomes to autosomes. Another important and interesting quirk of fruit fly genetics is that male Drosophila have no recombination between their chromosomes. In this video, I'm going to give a brief introduction to the Drosophila system, covering a number of important components, such as their life cycle, basic care and maintenance, crossing and genetic manipulation, and best practices for raising and keeping fruit flies. Okay, let's dive into it. Under optimal conditions, a fruit fly will go from an egg to an adult in about 10 or 11 days. These optimal conditions are 25 degrees Celsius and 60% relative humidity, with the flies being fed relatively nutritious food and existing at a low to medium density. So they're not being overcrowded, but they will also suffer if there's an extremely low number of Drosophila in the vial. This graph shows the basic steps in Drosophila development, from egg to the 3 larval stages, to their pupae, to the emergence of an adult. Fruit flies are typically housed in narrow vials with a block of semi-solid food at the bottom. Typically, the average carrying capacity of a vial is around 80 to 120 flies at a healthy density and healthy numbers. In a lab, the typical life cycle is that eggs will be laid on the surface of this food, and then the previous generation of adults that laid these eggs will be removed from the vials. After that, the eggs will hatch, and the larvae will burrow down into the food, eating and growing until they reach their 3rd instar. At this point, the larvae will crawl up the walls, form pupae, and metamorphose into adults. After a couple of days as pupae, the adults will emerge from their pupae, or eclose from their pupae, and then these adults will be transferred out of the old vials and into new vials, where they'll lay eggs and start a new generation, repeating the process. Fly labs use a number of different habitat containers to house their flies, from the ubiquitous narrow vial to larger bottles, or even very large population cages. Despite the name, fruit flies don't actually eat fruit. They actually eat yeast. So the standard Drosophila food is a mix of yeast, sugar, agar, some added nutrients, and a number of chemicals that act as fungicides and preservatives to prevent anything gross from growing in the food. The food, as I mentioned earlier, is mostly for the larvae, which will eat pretty voraciously as they go through their 3 instars. The adults don't eat that much, although females will sometimes eat a little bit of yeast when they're in the process of laying eggs. To encourage females to lay more eggs, you can sprinkle a little bit of live yeast onto the surface of the food, as seen here. Okay, let's briefly cover the major stages of fruit fly development. The first stage of fruit fly development is the egg stage. Fruit flies will lay eggs almost immediately after fertilization, although females will store sperm from a mating almost for their entire life, which they can use to make a continuous number of eggs without needing to be remated. A healthy female in an optimal condition will lay around 50 eggs a day on her own, although flies have been known to lay up to 100 eggs per day. Flies tend to lay their eggs in small clusters, either near the walls or on patches of particularly nutritious food. This graph in the bottom here shows the typical distribution of eggs, where the yellow is the food in the vial, and the eggs are clustered around the edges in a narrow ring. These eggs are microscopic and are very difficult to see with the naked eye, although you can just see them if you look very closely. Typically, eggs will hatch within a couple of hours and definitely within a day. When the eggs hatch, fruit flies emerge as larvae. small worm-like creatures. These larvae will go through 3 distinct developmental stages called instars. The first instar is almost microscopic, smaller than the egg, and they typically stay on the surface of the food and then will begin to burrow down into the food. The second instar is visible, but is very small. If you look at the top edge of the surface of the food, you should be able to see second instar larvae crawling around and burrowing into the food. The final larvae stage is the third instar. Third instar larvae tend to be very large, very clearly visible to the naked eye, and you'll be able to see them burrowing into the food around the edge, as well as beginning to crawl up the sides of the walls. Once third instar larvae have eaten enough food, they'll crawl up the walls and begin to form pupae. Fruit flies are holometabolists, which means they undergo a complete physical change from the larval to adult stages. Fruit flies will form cocoons called pupae or pupariums, where they will undergo this drastic change. Initially, the pupae will be very light brown, but as the flies develop, these pupae will darken and become increasingly opaque over time. When the flies are almost ready to eclose, the pupae will become visibly dark, and if you look carefully, you'll be able to see that the darkness is from 2 black patches, which are the nascent wing buds of the adult. Typically, flies will remain in their pupae for 4 to 5 days, and once you start seeing these dark pupae with the visible wing buds, you can assume that those flies will emerge from their pupae the next day. Fruit flies have a distinct circadian rhythm and tend to be most active at changes in light, either right around sunrise or right around sunset. As such, adult flies will typically emerge from their pupae at sunrise, which in a laboratory setting is when the lights in the incubator turn on. Right after they emerge, the freshly emerged adults will be very pale, very squishy, and very delicate. These unmated flies will have a visible meconium, or dark spot, on the underside of their abdomen. The adult flies will begin to harden after a couple of hours. After approximately 6 to 10 hours, these flies will mate for the first time after their exoskeleton has hardened completely. This is what a normal adult fruit fly looks like. Like all insects, they're divided into 3 segments: their head, thorax, and abdomen. If you look at the head, you'll see that they have 5 eyes, 2 large compound eyes, and 3 much smaller, simpler eyes called ocelli, in a triangle on the top of their head. These ocelli are used for sensing changes in light, and the flies use them to navigate in 3 dimensions. The head also contains the antennae, which in fruit flies are quite short and small, as well as the mouth parts. Fruit flies have 6 legs like all other insects, but unlike all other insects, they only have 1 pair of wings. The second pair of wings is greatly reduced in fruit flies and has been modified into a structure called the haltere, which is an advanced aeronautics organ which is used to sense air quality changes and used for advanced flight. The thorax, as well as the rest of the body, is covered with a number of hair-like bristles called setae. In fruit flies, the number, length, and position of the setae is very consistent and is determined entirely by the genes. There are several mutations which change the position or length of the setae, which are used as visible marker mutations, and we'll see some examples of these later. The last segment of the fruit fly is the abdomen, which contains the genital structure. In males, this will be a small, dark structure which looks a little bit like a black flower, whereas in females, this will be an extended tube-like structure. As mentioned earlier, fruit flies are sexually dimorphic, which means that there are clear and visible differences between males and females. In the males, seen on the top, they tend to be smaller with dark abdomens that curl underneath their body so that they're pointing either down or almost slightly forwards. Their genital structure, as I previously mentioned, looks like a small dark black flower. And if you look at their front legs, you'll be able to see small black structures called sex combs, which is a tight cluster of bristles that are used during mating. The females, on the other hand, tend to be larger than the males, with a lighter extended abdomen that tends to point straight backwards as opposed to curling underneath the body. As previously mentioned, they have a tube-like genital structure, and they do not have sex combs on their forward legs. Now we'll get into how to take care of fruit flies in a laboratory setting. The basic fruit fly maintenance schedule is a 14-day cycle, where you give them 3 days to lay eggs and then 11 days to develop, assuming that they're being kept at optimal conditions. This 14-day cycle allows you to flip the flies on the same day once every 2 weeks. It's also a very good practice to have a replicate population, so a complete duplicate of all of the stocks that you have, staggered by 1 week, so that you will flip all of your week 1 flies on 1 week, then your week 2 flies the next week, the week 1 flies again on the 3rd week, and so on and so forth. This 14-day cycle can be extended by keeping the flies at lower temperatures. For example, keeping flies at 18 or 19 degrees will extend this 2-week cycle to about 3 weeks. This sort of schedule is ideal for stocks, or basic strains of flies that don't need a lot of attention or maintenance, and you just need to try to keep alive long term. For more complicated stocks, or for actual experiments where you want to combine different genotypes of flies together, you'll need a more advanced and complicated maintenance routine, which we'll talk about now. Doing genetic research with Drosophila tends to involve a huge amount of genetic crosses, which is matings between different flies that have different genotypes in order to create an offspring that has a very specific genotype using a combination of chromosomes of interest from the parents of that previous cross. This is what a standard cross scheme looks like. These are the two parents, the males on the left of the X and the female on the right, showing the genotypes of their chromosomes at each chromosomal position. The leftmost set of chromosomes are the sex chromosomes. In the male on the left, they have an X chromosome with the white 1118 allele over the Y chromosome, which is that half arrow-shaped line. In the female on the right, they have two X chromosomes, each carrying the allele white 1118. White 1118 is a recessive allele that causes the carrier to have white eyes, as opposed to the standard wild-type red eyes. In males, since they only have one X chromosome and one Y chromosome, these recessive alleles will still be expressed since there is no good copy on a healthy X chromosome in order to cancel out the effect. The female on the right is homozygous for white 1118, so she will also have white eyes. The second set of chromosomes are the second chromosomes. In the male on the left, they have one chromosome that has CNBW spec and the DS red allele, whereas the second chromosome only has CNBW spec. In the female on the right, one of their chromosomes contains CYO, curly, as well as GLE, which stands for glazed. CN and BW stand for cinnabar and brown, which are both eye color mutations. Cinnabar and brown are both recessive, so if you are homozygous for them, it will change the color of your eye. Cinnabar causes the eye to be dark red as opposed to the standard bright red, whereas brown causes the eye to be predictably brown. Since cinnabar and brown each knock out separate pigments in the eye color pathway, a fly that is homozygous for both cinnabar and brown will have completely white eyes since both of the pigments that make up the color of its eyes are knocked out. SP stands for speck, which codes for a small dark speck next to the wing. DS red is an artificial fluorescent protein which causes the flies to glow bright red under the right spectrum of ultraviolet light. The last set of chromosomes is the third chromosomes. In both of these flies, they have regular wild-type third chromosomes denoted by WT. Here are a couple of the common genetic markers that we use to mark flies. On the top left is Oregon R, a wild-type fly which is the standard, normal wild-type fruit fly. On the right, we see a couple of the most commonly used genetic markers. Top left of this block of 6 on the right is CYO, or curly, which causes the wings to curl up and around. Next to it we have glazed, which changes the shape and color of the eyes, as well as yellow, which changes the body color from a dark brown to a very pale yellow. In the second row we see stubble, which causes the setae on the thorax to be much shorter and smaller. You can see in Oregon R that the setae are long enough that they extend a ways beyond the thorax and cross above the abdomen, whereas in stubble they only extend a short amount and do not cross. Next to it we have white, which is an incredibly obvious mutation, which causes the eyes to be completely white. And lastly we have DS red, which is an artificial fluorescent protein, which causes flies to glow red under the correct spectrum of light. Here DS red is being expressed in the eyes primarily, which causes the eyes to grow incredibly brightly red. On the very bottom left, we have ISO1, which is a very common laboratory strain, which was used for the original sequencing of the Drosophila genome. You can see that it has a number of mutations. It has yellow, causing its body to be a more pale yellow than Oregon R, as well as CNBW, which causes the eyes to be completely white, as well as speck, which creates a small black dot right at the point where each wing meets its body. There are many different applications for these visible mutations, but the most common and most powerful application is as part of a balancer chromosome. In a normal chromosome, all of the DNA is oriented in the same direction, which allows recombination between similar chromosomes. In a balancer chromosome, huge inversions cover all or most of the chromosome. These huge inversions prevent recombination as the DNA is oriented in the opposite direction to the other chromosomes. In a balancer chromosome, this huge inversion contains a visible dominant marking mutation, as well as mutations that make the balancer chromosome homozygous lethal. There are an enormous amount of uses for these balancer chromosomes. You can use them as an intermediate step in a cross, as you know that any offspring from that cross that contains the balancer chromosome will be heterozygous for the balancer chromosome and your chromosome of interest, since the balancer chromosome can never be homozygous due to the homozygous lethal mutations. You can also use a balancer chromosome to maintain a different homozygous lethal mutation in a stock over a long period of time, since the only viable offspring of an individual that has a lethal homozygous mutation in a balancer chromosome is an individual that is heterozygous for both the lethal mutation and the balancer chromosome, allowing you to maintain the stock almost indefinitely. These visible mutations in balancer chromosomes make setting up crosses between different strains relatively easy, but there is one annoying peculiarity of the Drosophila system that you need to consider. A female Drosophila can store sperm from a single mating and use it to fertilize eggs for days or even weeks after that mating. So in order to ensure that the products of a cross are what you expect them to be, you need to collect unmated or virgin females to use in that cross. There are two different ways to collect virgins, either collecting them as adults after they've emerged from their pupae but before they're able to mate, or collecting them as pupae before they emerge. So flies tend to do everything around changes in light, whether that is mating or laying eggs or emerging from their cocoons, and typically they do this in the morning at the change from night to day, as opposed to the evening, although they are still active in the evenings. So when collecting virgin adults, you need to go in before the change in lights, so before the lights turn on. Clear any adults that have previously eclosed out of the vial so that the only thing in that vial is pupae. You need to be really, really careful to clear out every single adult, as all of the adults that are in there are typically going to be old enough that they're capable of mating. After you've cleared out all of the adults, wait for a couple of hours so that the lights change and the flies have a chance to emerge, and they have a chance for their exoskeleton to harden enough that they're not as incredibly delicate as they are right when they emerge. It will typically take around 6 to 8 hours after emergence for flies to mate for the first time. So all of the flies that emerge within that window, that 6-hour window between you clearing out the adults and them mating for the first time, should be virgin. You should still, however, check to make sure that they are virgin by placing the females alone into a vial and leaving them for a couple of days to see if they lay any fertilized eggs. Females that are unmated will still sometimes dump eggs that are unfertilized, but these eggs will never hatch into larvae. So you want to wait a couple of days and check to see if there are any larvae in the vial, not eggs, larvae, because the presence of larvae shows that some of the females in there have been mated with and cannot be used in the experiment. You can also collect virgins as pupae. Collecting adults takes a large chunk of time during the day, and sometimes this is inviable. Collecting pupae is a much faster process in that it takes less time during the day, but it's a more labor-intensive process than collecting just adults. So what you want to do is take a wet paintbrush and use it to gently pull the pupae off the sides of the vial. The water will dissolve the glue that holds them to the side of the vial, and then they'll stick very slightly to the paintbrush, which you can use to lift them out of the vial and place them on the flow pad. Once they're on the flow pad, you can use the microscope to sex the pupae and separate females from males. All males have sex combs on their front legs, which is this cluster of thick, dark bristles, which is actually visible through the pupae. So all males will have visible sex combs inside their pupae, and all females will not. So if you look for the sex combs, you'll be able to separate pupae that contain males from pupae that contain females. You can then use that damp paintbrush to stick these pupae onto the walls of a new clean vial. So you'd put female pupae in one vial, or it's best practice to put female pupae in a number of vials. That way you lose fewer if you have a contaminated vial. And then you can place all of the male pupae into a single vial or a small number of vials, whichever you prefer. So doing all of this requires some equipment. Fruit flies tend to fly, which makes it relatively hard to work with them while they're active and awake. So whenever we want to manipulate or look at or phenotype flies, we have to use some method to render them inactive. There are a number of different ways to do this. Flies will become inactive if they get cold enough, so you can place them over ice. You can also use chemicals like ether or some commercially available chemicals like FlyNap or carbon dioxide. In our lab, we use carbon dioxide. We use a blowgun to dispense carbon dioxide into the vial, and then once the flies become inactive, you can put them down onto the flow pad. The flow pad is a porous plastic block with carbon dioxide running through it, so as long as the flies are on the flow pad, they will remain inactive. If you leave them there for too long, they will eventually begin to suffocate, and the carbon dioxide will begin to become stressful for them and can cause health issues down the line. So as a general rule, you don't want to keep your flies on the pad for more than 10 minutes at a maximum. This is an image of my current FlyWorks setup. In the center is the dissecting microscope that I use to look at flies and phenotype them. On the top left, we have the light box, which is the light source for the microscope. The large knob on the right of the light box turns it on and controls its intensity. Next to the light box, we have the morgue, which is a flask full of 70% ethanol, which we use to dispose of unneeded flies. Next to that, I have my flask of ethanol, which I use to sterilize equipment and surfaces when I'm finished with them. Looking at the surface of the microscope itself, I have my paintbrush, which I use to manipulate flies, as well as the CO2 flow pad, which I will place my flies on when I'm working with them. Next to the flow pad is the blowgun, which I use to inactivate the flies in the vial so I can take them out. Both the flow pad and the blowgun are connected to the CO2 bubbler, which is a flask filled with water that the CO2 is bubbled through to humidify it. Humidifying the CO2 prevents the flies from desiccating or drying out while they're on the pad, and it also reduces static, which makes it much easier to work with the flies. Finally, the whole CO2 system is connected to the CO2 valve, which controls the CO2 system. You want to be careful not to open the valve all the way, as that will increase the pressure so high that either the cap will blow off of the CO2 bubbler, or the pressure in the blowgun will be so high that you will tear the wings and legs off of the flies, which obviously is quite harmful to them. So you want to make sure that you have a gentle enough pressure that you can put the flies to sleep without damaging them or damaging the system. Once you have the flies inactive, you want to carefully dump them out onto the flow pad, which you can do by keeping the vial on its side as you insert CO2. If you keep it upright with the food at the bottom, the inactive flies can fall onto the food and get stuck onto it, as it tends to be somewhat gooey and sticky. So you want to keep the vial either on its side or upside down while inserting CO2, and then you can gently pull out the plug and tap the flies down onto the flow pad without banging the vial onto the flow pad directly, as that damages and dents the flow pad and can prevent CO2 from bubbling out of it. Once you have the flies on the flow pad, I like to spread them out into a thin line, and then you can move from one end to the other, looking at each fly one at a time, phenotyping it, and then using your paintbrush to gently move it either up or down depending on what phenotype it is. Once you have all of your flies separated, you can easily brush flies off the edge of the pad into a vial, and then brush any flies that you don't need into the morgue. There are a number of different traits that you can phenotype flies for, the most common of which is sex. So looking at this image again, you'll see that there are a number of incredibly obvious sex differences between male and female fruit flies. The males tend to be smaller and have very dark black abdomens that curl under their bodies so that their genitals are pointing either down or slightly forwards. Females, on the other hand, tend to be much larger than males. They have pale abdomens, and their abdomens tend to extend backwards, ending in a point. The size and coloration of the abdomen are not always going to be consistent, and you cannot rely exclusively on color and size, since flies tend to vary in size and coloration, typically dependent on their health. So less healthy flies will be pale, and all of them will be equally small, which makes it very difficult to separate males from females. You can always be sure that you're separating flies accurately by looking at their genitals and their sex combs. In males, the genital structure looks like a small black flower, whereas the genital structure of the female is an extended tube coming out of the end of her abdomen. Also, if you look at the front pair of legs, you'll see that males have a thick, dark patch of bristles called sex combs on the insides of their front legs, whereas females will not. All males will always have sex combs and a genital structure, and all females will not. You can phenotype all of the other visible genetic markers the same way you would phenotype sex. Spread the flies out into a thin line on the flow pad, then use the paintbrush to separate the flies that have the phenotype you're interested in from the other flies. Place the flies you want into new vials and dispose of the other flies into the morgue. That pretty much wraps up basic fly care. I'm now going to cover a couple of general best practices and methodology for making sure that you're doing the best fly work possible. Issue number one is contamination. As I previously mentioned, flies fly, which means they get in everywhere. So it's very possible and a constant risk that a fly with a genotype that you don't want will somehow find its way into your vials and can then mate with or lay eggs in those vials, contaminating the genetics of whatever cross or experiment you're trying to do and ruining your entire results. So you want to be very, very careful to do everything you can to minimize how many flies escape. So when you're flipping or transferring or clearing flies, you want to be very careful so that you don't slip up and let flies escape. When you're working with flies on the flow pad, you want to make sure that all the flies end up on the flow pad and that you don't lose any when you're working, or you don't lose any when you're putting flies into the morgue or into the vials, as those flies will wake up relatively quickly and then fly away. There will always be some number of irreducible rogues flying around in the lab. So whenever you're working with fly food, you want to keep it covered as much as possible, which means keeping the plugs in vials for as long as possible and only uncovering them when you're about to fill them and then immediately covering them up again. You also, whenever you use a vial or are about to fill a vial or a bottle or a cage, you want to check to make sure that there's no rogue flies already in there. Even if a fly is only in there for a second and then flies out, it's possible that they may have laid eggs, which will then hatch in that vial and contaminate your results. So you want to be very, very careful, and just as a general practice, don't use any food that you see a rogue fly get into and out of. And whenever possible, if you see a rogue either flying around or crawling on the counter, you want to squish it to reduce the amount of rogues that are flying around in the lab. Another huge issue is labeling. You want to make sure that everything you work with is labeled, and clear and accurate labeling is critical. It's pretty common for labels to become really messy or involve a lot of shorthand or use symbols that only you can understand. And these are all pretty bad practices because it makes it hard for anyone else to use your work. And also, if enough time passes, you'll just forget what that combination of symbols is supposed to mean, which happens to me all the time. So you do want to make sure that your labels are as clear as possible, as unambiguous as possible. So you want to make sure that your letters and numbers are all very clearly recognizable as exactly what they're supposed to be, and that you and anyone else can always read them and know exactly what's supposed to be in that vial. You also really do want to label literally every container that has a fly in it with, at the minimum, what strain or genotype that fly is and what date that fly was put into that container. And if you have tape labels that are starting to become faded or damaged or otherwise unreadable, you want to replace them as soon as possible, ideally before they get so faded or damaged or unreadable that you can't accurately copy down what's on it. If you look at this example, it's a very cramped, messy label that I've rewritten to be unambiguous and clear. You also want to be very careful about sterilization. It's possible for flies to contract and spread infections, whether it's viral or bacterial or fungal or parasitic. And any bacteria or fungus or mold that gets into the food will outcompete the larvae and kill them, which will ruin your experiment. This has happened to me before, where I've had bacteria absolutely destroy a huge amount of work that I was working on, and it was a terrible experience. So you want to be very careful. And being very careful is pretty easy, luckily. So you want to make sure that you're keeping everything clean. So don't let mess pile up. If you spill food or any edible or rottable substance, you want to make sure that you clean it up promptly. No matter what it is, if you're using equipment, you want to make sure that you're sterilizing it afterwards with ethanol. So you should use a Kim wipe to sterilize your flow pad and the blowgun and the paintbrush when you use it. You should use ethanol and paper towels to sterilize the surface all around. Any surfaces that were in contact with flies. And you want to make sure that you're not leaving flies on old food for too long, as that food will begin to rot and spread infections and any bacteria that grows in that will be transferred to the new food when you transfer the flies over. Quickly, organization in general is hugely important. You want to keep the lab as clean and organized as possible. So you want to make sure that you understand where things belong, since most things do have a dedicated home where they live. So you want to make sure that if you take anything, you return it where it's supposed to be, and you understand how the lab is organized and where things are supposed to be. In general, just as a best practice for everything, you want to keep your workspace as organized as possible while working. Don't let it get too cluttered, and clean it up when you're finished. Another very general point is with fly work as well as all science, you want to be as consistent as possible, and you want to pay as much attention to detail as possible. These two things are really foundational to all science, and the more effort and attention to detail you put into the research, the better the research will be at the end. Making sure you're being consistent and making sure you pay attention to detail will result in consistent, reproducible science that has minimal bias and minimal noise in the data. And in general, the more effort you put into it, the better science you're going to get out of it. And as a sort of follow-up to that, you do want to think a little bit about ethics. Fruit flies aren't subject to the same protection that vertebrate animals like fish or mice are, but they are still animals and they are still living things. So you should treat them with care. You should treat them with as much care as possible. You really do want to give them the best treatment you can, which is the most nutritious food, the best living spaces, the most optimal conditions possible. So you want to keep your flies as healthy as possible, treat them as well as possible. And really, the healthier you keep your flies, the better the data will be. The healthier you keep your flies, the easier it will be to phenotype them, the more quickly they'll go through generations, so you'll be able to do science either more quickly or you'll be able to get more done. They'll be more consistent, they'll be more predictable, so unhealthy flies will take longer to pupate or longer to develop or longer to lay eggs, which will throw off your whole schedule and be a huge pain. So the better you treat your flies, the better science you'll get out of it. But you shouldn't just take good care of them for the sake of science, although it's a huge factor. You should take care of them because they are living things and they deserve respect. So you should never do anything unnecessary or cruel to your flies. And you will need to dispose of a large number of flies throughout the process, but you want to make sure that they're disposed quickly and effectively so you're not doing anything that will cause them to suffer for any length of time. So that wraps up my short, this is like 30 minutes long now, short introduction to fruit flies. There are a number of good references that I find myself referring to a lot. The University of Washington has a great introduction to fruit flies where I got a lot of these images from, which you can find at this link. Ralph Greenspan has a Drosophila Bible called Fly Pushing, which is mostly about really hard genetic material, but there is a great couple chapters on caring for flies and setting up crosses. I have a copy that I bought myself, and it's a good resource to be able to look at. And this article, The Life History, Evolution, and the Genetics of Fitness Components in Drosophila, is an article in the Genetics Fly book, and it's just a really good resource, especially the first half, for understanding the life cycle of Drosophila, and a lot of my earlier material is drawn from this article. So I recommend, if you want to learn more about flies, that you check out these 3 sources, and thanks for putting up with me ramble about flies.